Hello, my name is Mike Raj, and I will be presenting about acids, and this is for GCU class concepts in biochemistry. And these are two of my colleagues that work with me to finish the PowerPoint, Melinda and Rebecca. And my name is Mike once again. So, what are nucleic acids? These are one of the four major classes of biomolecules found on Earth. They are also known to be macromolecules, larger molecules. So, what are the basic units of nucleic acid or the elements? The elements contained are carbon, hydrogen, oxygen, nitrogen, and phosphorus. Because carbon is so ubiquitous, that's why it's called organic molecule. They are all macromolecules. They are made up of single units called monomers that are joined together by covalent bonds to form polymers, which are large molecules. So how is this macromolecule built? It is through the process of dehydration synthesis. Occurs when a monomer joins to another molecule to form a covalent bond and releases a water molecule. Here, as you can see, two molecules are joined, and uh, hydroxy and hydrogen is removed in the form of water, and these two molecules are joined and the ether formed, aka condensation reaction. It typically requires a high amount of energy investment. So, how is this molecule broken down? The molecule is broken down through the process of hydrolysis, and in the process of breaking down bonds, energy is released. The main molecule uh, to break down these bonds is water, H2O, and here in this ether, it is attacked on the either of the sides and it's broken down. Occurs when a polymer are broken down into monomers by addition of water molecule for each bond broken. So it's this process called hydrolysis. Nucleic acid monomer, nucleotide. So what's the basic building block of a nucleic acid monomer? It's called a nucleotide. It has a nitrogenous base, 5 carbon sugar, and phosphate. Phosphate is the backbone which can't be replaced by any other element. One or more phosphate groups, pentose, it's a 5 carbon sugar, either deoxyribose or ribose, and nitrogen containing base, nitrogenous bases or nitrogen containing bases. Include the following in the DNA. So the DNA and RNA, the only difference is ANU. So the A stands for adenine, T thymine, C cytosine, and G guanine. And for RNA, the adenine is replaced by uracil. And there's different categories of purines and pyrimidines. The pyrimidines have a single ring, cytosine, thymine, and uracil, where purines have guanine and adenine, which have double rings and closed rings. Different classification of nitrogenous bases based on their fundamental structure. The moiety is kind of different, as I mentioned before in the previous slide. So the structure of DNA, it's known as a double helix, describes how strands of DNA are wrapped like a spiral staircase. Functions like a ladder, information ladder, alternating sugar and phosphate sides. The phosphate is the backbone which holds together the nitrogenous base and the sugar, the sugar ring, the five-sided sugar ring. Alternating nitrogenous base rungs of ladder. The nitrogenous bases, the complementary, actually contains information. That itself is information which is used to build up to build up to build up the biological information. The structure of a DNA bases are connected to the first carbon Phosphate groups connect to the 5 carbon prime. Here's the 5 carbon prime, and here's the 3 carbon prime. So it's complementary. And the two strands of DNA are antiparallel in nature. So the 5 prime to 3, so the opposite would be 3 prime to 5, and they match. And the nitrogenous bases has to has will have to go with A adenine goes with tyrosine and guanine goes with cytosine. But if it's RNA, then the A is replaced by uracil. That's the major difference between RNA and DNA. Structure of RNA single-stranded. Structure is similar to DNA except the uracil. Concept of alternating sugar phosphate backbone, uracil instead of thymine. mRNA, small travels, less stable, only codes for a small portion of the entire genome, carries the code to make protein. So the basic purpose of mRNA is to carry codes, the information codes in small strands from the nucleus to the cytosol. DNA large remains in nucleus, more stable, genes-based sequence replicated to make new cells. Another interesting fact is DNA is never completely unwinded. Only a small portion is unwinded at any given time, while RNA is single-stranded for easy replication. Protein synthesis is the central dogma of biological science. So the process goes from DNA to RNA to protein. Protein is the functional building block for life. And the other aspect is from DNA to RNA, the process is called transcription and happens within the nucleus. And for the second process, from RNA to protein, it's called translation, and it happens in the cytosol, as you can see in this slide. So what is a protein? Protein is a macromolecule made of amino acid chains. Um, Amino acid chains. There's a couple of amino acids which we didn't go into detail here, but these amino acids are a building block by which protein is made. So there are three and RNA involved in this process of protein construction: mRNA, which is messenger RNA; tRNA, which is transcription RNA; and RRR, ribosomal RNA. So the beginning to end time depends on the kinetics and the availability of RNA. It's quite quick, mere minutes to hours. And these proteins are eventually degraded by lysosome. It's a vesicle which proteins are moved in and broken down by strong enzymes. Proteins make up the functional biology, from enzymes, cell membrane to hormones, and it's called proteolo- proteogenomics, I believe. It's hard to pronounce. Proteomics. It's a science specifically dedicated to the study of proteins. DNA paper modeling activity. So here you can see students should work in pairs, provide students with pre-cut components, have students construct a nucleotide, label the deoxyribose sugar carbons, label purines and uh, pyrimidines, and so forth, so forth. I apologize, there was an error over there. Mm. And the other one is POGO, which is process-oriented guided inquiry learning. You have to pay for it. It helps students to understand more in depth by providing them with activities. It's basically oriented towards mm, as a auxiliary guidance worksheets and so forth. And here's the references. And once again, this is my crash lecture on nucleic acid for GCU class concepts in biochemistry. Thank you. Have a nice day.